Welcome, everyone. Happy Halloween. It's great to see that a few people got into the Halloween spirit, including our speaker. So it's going to be a very fun presentation. As a reminder, we have an online audience in addition to everybody in the room. So if you are online and you have questions, please put those in the Q&A box and we will be monitoring that and can pass those along to our speaker. If you're in the room and you have a question, of course, just raise your hand like you normally would. But if you can try to speak up a little bit, that would be helpful to make sure that our online audience actually hears the question in addition to the answer. I think that's actually all I have in terms of announcements. I'm pleased to present or introduce today's speaker. We have our construction worker here, Hsiao, who is a PhD student in DCMB.
Hi everyone, I'm Hsiao, or you can just call me Leo. Today I'm presenting part of my project recently published. It's called Analysis and Visualization of Quantitative Proteomics Data using FragPipe-Analyst, and I'm currently fifth year PhD student from University. Yeah, and I feel like people might wonder why I dress up here, probably like construction worker or like minion, but I feel like that's kind of around the spirit of our tool. So this is our lab's tool called FragPipe, and this is me like right now presenting our lab's tool. I feel like a general like construction thing or like pipe thing. But anyways, like happy Halloween. I hope people can enjoy the presentation and let me know if you have any question. I think I'm pretty okay if you want to interrupt me at any time to figure out any kind of not clear part. So just a step back, that overall goal of the university lab is focus on mass spec based proteomics analysis tool. At the same time, we perform many large-scale proteomics analysis projects. One particular example I involved is this clinical proteomics tumor analysis consortium. And another step back is I figure people might not understand what's a mass spec-based proteomics that much. You might hear transcriptomics a lot, but proteomics is a kind of omics data like closer to a phenotype, so it's also gaining much more interest. And there are more than one way to do a proteomics, but the topic I'm going to focus here will still like a mass spec-based proteomics. I think that's an even older technique compared to some people can do like single cell and use antibody to kind of profile the protein computation. But in that strategy, I think right now the throughput is still like 2,000, 3,000 protein in one time. But mass spec generally we can get like 10,000 in general, and it's like a lower limit you can get. But the general principle is when you have a protein sample, you will digest it into a peptide because it's a kind of a unit we can quantify, and then go into an ion source, and this ion source will put those peptide ion into like mass spec, and we use like more than one mass analyzer to really get a protein quantification. Those peptide ion will go to a mass analyzer one and further fragmented and put into like second mass analyzer, and then we get a detector, and that's what we call the mass spectrum. And generally, we will also couple with liquid chromatography to do the sample separation, so we can only get a small number of a protein in one window. And this will be collected in the MS1. MS1 is a peptide ion, and then go to MS2. This peptide will get further sequence, as you can see in the lower panel here. And on the laser pointer, I feel like this might be easier to see where I'm pointing. Yeah, and recent year there are many breakthroughs in this area. I think if you start probably like 20 years ago, I think you will know that proteomics database through mass spec, they have not missing value, but we have more and more either from the instrument or algorithm part, we are able to do a quantification better. But generally, just as an introduction and background, since this one of the platform and we'll talk about later in the presentation, this TNT or tandem mass tag is one of the techniques that can help us to like do the protein identification and quantification at the same time, and also like since it's a technique like mix many samples together, and you can get a quantification in one. So it also reduce a little bit of the batch effect if you want to like run sample separately. So, but the general principle is you have a kind of label things. So in general, this kind of technique called label base, compared to the earlier version, probably the original version, we call it like a label free. And then what's kind of label being added to into a sample is kind of like this chemical structure. And then since this chemical structure is well designed, when it go into MS1 and MS2, they will kind of separate apart, and you can use one of the group as the quantification value to reflect how much protein you have. So, but it's just a general introduction of this technique. The other technique I want to talk about is when you think about how we do a protein quantification. Usually we talk about an older technique called data-dependent acquisition. What that means in the MS1, we will only select part of the spectrum that with really high intensity and then go to MS2. But since the instrument breakthrough and also like lots of different techniques and instrument design overall, I think right now another way of doing this is rather than actually seeing a small window and higher peak, we can use all of the window, all of the MS1 peak. They cause some problem. For example, you might have more than one protein contained in this isolation window, but overall this technique also have a better protein identification and quantification. And no matter which technique you are using, computers still kind of play an important role here, since you can see the complex data structure. You have MS1, and every MS1 you will derive several MS2, and you need to have lots of different algorithms by informatics tool to do the analysis. So FragPipe, as I introduced in the beginning, we have been working on this for several years, and this software also being used by many people. I think we can kind of confidently say we take probably around like 20 or 30% of the user in this field that we're using our tool. And generally, we have this FragPipe ecosystem, but when we're reviewing our current strategy and what we are missing, we realize that most of the user will find it's a little bit difficult to use some of the things because as an academic lab, we develop to one by one, and some of them like developing by several different people. So one of the key steps is the quantification part, and this actually come from like 4 different projects. So various of the output format from the quantification step become a part that many users will get confused when they switch a platform. They feel like it's not user friendly. And as I mentioned, TNT is another strategy. It reduces the batch effect, but generally the statistical property of the TNT data will become different from like the LFQ. They don't able to quantification or DIA still label free, but generally the statistical property are also different. So if you are a beginner, you might make some mistake in the beginning. Well, probably the most important part as a bioinformatics students is I find lots of the paper when they report something, even I want to reproduce is a little bit hard. So generally, in my mindset, it's still good to have a kind of tool can address this issue. And so we are missing a one puzzle here that can put all of the pipeline together and being really useful. And at the same time, during my kind of like 4 to 5 years here, I also involved many large scale cancer proteomics project. For example, this CCRCC published last year in the Cell Report Medicine is actually based on another cell paper published several years ago by our lab. We have extended the cohort and include more different kind of a tumor and perform like different kinds of a PTM together to do a quantitative analysis. The other project I have is like non-adenocarcinoma. It's an intentional collaboration project with Taiwanese team. So overall, this kind of a collaboration usually involves more than like at least like 20 or 30 people a time. I feel like it's really hard to even like communicate, saying that hey, I want to do this. We can think of like more than 3 ways or 4 ways to do analysis. So if we can have a common language in a sense of a software, then probably we are more possible to reproduce our result. Those papers are pretty big, but when you want to reproduce it, you send an email to a corresponding author, you realize, no, this every panel probably come from different author. Let's just really hard to reproduce all of the stuff. So we investigate a little bit like how many different software out there and what's the existing software, and for publication and also probably just general academic thing is we want to find some unmet needs that we can improve. So we realize lots of people still use like old technique like label-free quantification, but other software only support this, and we want to support others like TNT and DIA, as I mentioned earlier. And the field also, since proteomics is a technique kind of different from the RNA-seq, the experimental design really depends on like what kinds of readout you want. You probably want to focus on some of a residue of a protein or even a peptide. So some of the recent trends is, are we able to do a peptide level quantification and also like do analysis at the peptide level? Because when you use MS1 and MS2 to do like protein quantification, there's actually a step called protein inference that will take a peptide, but those peptides might not just only map to one protein. So the assignment of the protein is actually an ambiguous thing, and sometimes we know it might make some mistakes. So some people will feel like probably go back to a peptide level and decide any kind of assay at the peptide level might be of the interest. So peptide level quantification is something we want to incorporate. Another kind of a change is lots of protein will go through post-translational modification. They are really site-specific. We want to look into like different mechanisms. For example, phosphorylation is one particular mechanism. Only particular amino acid residue will be the focus, and every protein they have many residue can be phosphorylated. So we really want to do some analysis at the site-specific level. So here is a kind of replicating, but it just summarize what we observe and being the objective of our project. So overall, from this publication that we recently published, we kind of come out 2 different tools. Since this is a tool and tech seminar, I want to mention this in the beginning. We have 2 different tools out of the publication. One is the FragPipe-Analyst. Is the major thing I will discuss today is a web interface. And as a bioinformatics student, you might have much more programming experience than FragPipe-Analystr. Is a more advanced tool. It's a R package. You can use it. I think that's generally our recommendation. And both projects are open source, and we are still active developing it. So if you have any requests, you can contact us, and we can fix about those things together, even have a collaboration. So another thing I want to highlight before jumping into the research detail, I really like this kind of thing, since the kind of follow up with like my learning experience in the DCMB is a tool that we kind of build and test across many different DCMB classes, and now is with much more attention. Originally, it's kind of like a side project. I just noticed this one paper. I kind of like how they are doing, but I want to improve that. And later, I present those things in the lab, and we find, wow, it's actually an interest in the lab that we probably want to incorporate. So I start doing some prototype, and then even like, since we have some students taking our class on the proteomics part, so we release this prototype to students, and students can test. And then further, since as a bioinformatics student, I also want to do more coding a little bit. So in the class, like instructed by the instructor before is like 576 is a package development. So I put this project into it. And then further, even like this semester as a GSI, I also like release another kind of a new feature and can be tested in the homework. And this tool kind of been highlighted in several different ways in the field. We kind of get one probably most popular unofficial blog post on this one, and also from the journal, is the most read article in the past 30 days. Yeah, so back to the topic. FragPipe-Analyst, just as an overview of the FragPipe-Analyst, it only takes 2 input. It's not really complicated, but we want to make it as accessible as possible, because when people are looking at data, they might come from a clinical field. For example, my collaborator, some of them are doctors, some of the pathologists. They find it really hard to just work on the Excel table. They might have like some knowledge, but if you have an interactive platform, then it's easier for them to go through a thing. So generally, they have 2 inputs, so quantification report, and the other is the experimental annotation. And here is the general fixie of overall pipeline view. We take those things together, and since proteomics data have lots of missing value, so we sometimes want to do missing value filtering, only keep some of the features you have confidence, and you can do a normalization, even like do an imputation. Imputation is a kind of, I always say it's a double edge. Sometimes it gives you good results, sometimes it's just you introduce some artificial thing into a data, so you need to very, very careful. But in terms of have just a straightforward pipeline, we still put in there, it's optional. And then those things, after doing those things, we can do a differential expression analysis and then further enrichment analysis. And not specifically like a sequential rule here, quality control and heat map can also being applied together. And generally, this kind of FragPipe-Analyst is implementing our Shiny app, so it's web accessible, and we can host it if you have a web server. And we are actually kind of expanding this infrastructure to see if we are able to help as many users as possible. And so here is another detail of the contribution table. Contribution table, if you are more familiar with the transcriptomic data, it's not really different. It's just a protein multiplied by sample metrics. So generally we will have some header like protein name or some additional metadata like a different database ID. And the experimental annotation is something more important, since sometimes like programming tool or like doing a downstream analysis, you want to name the sample differently. We have a sample name column, and we also have a condition. That's a major comparison, since lots of projects are involved, like CPTAG mentioned earlier, like cancer project, we focus on like what's the difference between the tumor versus like their normal adjacent tissue, or even like compared to different kind of tumors. So we want to set out the different condition. And replicate column is another way to like import the sample. Some people will kind of have a replicate in the cohort, but in this game sample, I didn't have many replicate, only one here. But generally, this is kind of another column. When you kind of do a visualization, you want the user to switch back and forth to see if they have like batch effect across the replicate. And to demonstrate the functionality of a FragPipe-Analyst, we kind of find several high-profile journal and find their data interesting, and we put it together, and they rely on a little bit different experimental design. And so we think it's also a good introduction to people, even like a beginner of a proteomics field, they might learn a little bit more about like mass spec can also be used to probe a different biological question. Here is a overview of those data set. It can basically divide into a 5-part. I will introduce every part later, but the general idea here is I want to mention is that many kinds of consideration in our tool design we've been covering in this example. And I will start with the CCR-CC1. This is the first cell, one of the cell manuscript we published, and they have 3 different examples here, but I will go through 2 of them first. But just general idea, this is a cancer cohort. So we are studying one major kidney cancer type called clear cell renal cell carcinoma. And in this cohort, we have some tumor and also normal adjacent tissue and other kind of like quality control thing. We incorporate several QC sample into it. And then the data was quantified by TNT and DIA together. They have a pretty complex workflow with it. So it could, I can collect part of a sample in the beginning using DIA, and the rest of the sample will be used to have a higher depth of the protein coverage using TNT data. And in this workflow, we also able to profile some of the PTM, and here is a phosphorylation. So I will start with a basic example. It's still doing a global proteome quantification and collecting TNT, collecting in the TNT data. So we just choose a subset as a demonstration. We take a subset that's around like 4 out of 23 templates experience, and we put it together, do a search, and after you get those report, we annotate a little bit of the experimental annotation, and we can submit to a FragPipe-Analyst. And then in the FragPipe-Analyst, we will able to see some of the analysis being popped up. One of the example is the PCA plot. So in the PCA plot, we are able to, like, it's a principal component analysis. Usually we will use this to kind of investigate if you have a batch effect or you have a expected separation on your like protein quantification result. But one drawback is in this principal component analysis, you expect your metrics need to be like complete. So you cannot really have any missing value. So that's one of the drawback. So, but at the same time, if you do it like a correlation heat map, you can use a pairwise correlation. So if the sample still measure particular feature between the two different sample, you can still try to calculate their correlation together. So it's another way to do the quality control. So as a result here, you can see the QC sample and tumor and NIT, or I can just simply call it as a normal. They have a well separate result, even like a correlation heat map that could consider almost all feature you have in the matrix still shows that they are separate together. So generally it looks pretty good as a demonstration of data quality of the experience. And then further investigation, one particular example we can demonstrate is in this kidney cancer cohort, we actually accidentally collect some of the kidney cancer, not a clear cell, but is the other subtype. So clinical people, they have their own usage of a biomarker. Probably they are really well known, so they will investigate a bit to kind of retrospectively to see if they are kind of like being like annotated wrong in the beginning. So here we also have a ability. You can just choose several biomarkers like well-known, well-defined in the field and looking into it. So here is an example like PIGR. We find there's one sample in this fourplex. They have like one example that is seems not like a clear cell, and you can also visualize this through the heat map. You will notice there's one example here like shows like a little bit different pattern. So we are able to demonstrate in this procedure, you are able to find some outlier and they can support some of the cosmic analysis you want to perform on your data set. And after removing those QC and potentially not clear cell renal cell carcinoma sample, then the first straightforward biological question you will ask is how much difference between the tumor and NAT. So if you annotate those samples correctly, this Pokémon plot will also be show up. So you are able to see some of the things difference comes out between tumor versus NAT comparison. And through this differential expression analysis, we can also perform the enrichment analysis based on like upregulated genes or downregulated genes and perform the overrepresentation test. And overall, the message we want to say is if you go back, check the cell manuscript, they report several findings. For example, tumor NIT, they have a well separation. They have a particular biological pathway being kind of like difference, or some of the protein marker they found of the interest. All of this, we can use a subset of the sample, and we provide like a tutorial thing. People can reproduce, just use the subset of the data, and generally speaking, they are really consistent. And we are able to show that you can use this tool to generate some of the plot that been published in the earlier cell manuscript. And since I mentioned TND earlier, and on this cohort, we also have the DIA data. So we also try a little bit to see if this DIA data is useful or recapture the sensing. And we perform the same analysis. And here is just an example. I pull out the PCA plot and the correlation heat map. They are generally pretty similar to a TNT, but just as a reminder, DIA and TNT, they are different waveforms. So people generally think that quantification doing by the DIA is much reliable compared to TNT. TNT will have like ratio compression. If you have a further question, we can discuss later. But here is another slide showing like those things we are able to reproduce. So right now I covered the first two example. Here I need to like jump a little bit far away from the clear cell renal cell carcinoma cohort. I'm going to introduce a different kinds of a mass spec technique. So APMS. APMS is a affinity purification mass spectrometry. It's a common strategy to profile the protein-protein interaction. And how that works is you will have an immobilized base protein being like lock on a surface, and then you will use your protein mixture matrix to flow through this like column thing, and then you are able to capture some of the interaction will be stay in the column, and you can take those samples and go to further digest and do the mass spec experience. And so experience. So this way is another is kind of to profile the protein-protein interaction, and they have many statistical procedure to be proposed to study this question. Even for our lab, we have kind of further tool to do the APMS analysis. But we are also interested to see if this FragPipe-Analyst is able to reproduce some of our results. So we choose this paper published in I think it's in Science. It's a label-free quantification APMS in background data. I look into a head and neck cancer and looking at one particular driver in this disease. So we take a subset of this driver experience and do a reanalysis. So go through a similar procedure. You upload the protein quantification table and also experimental design. We are able to generate those volcano plot, and those upregulate part. That means they have more interaction between bait and those your sample protein in the sample shows up in the upper right part. And this result is kind of consistent with if I apply a well-established tool published by our lab earlier. They are pretty consistent candidate. You can see like CK1B or 1C, those things, and CK2, they will co-consult. And if you go back to find like their original science publication, this example also illustrated. So generally speaking, even like by simple procedure, we are able to reproduce some of analysis in the APMS experience. And one consideration when you use this APMS protocol is this APMS protocol usually is label-free, so they have lots more missing value. And people, in one way, like the volcano plot shows here, we use imputation data, but some people will also feel like it's not safe. And generally, and I will also say you need to investigate a little bit to see your imputation doesn't influence your conclusion on the data. So as a way to support this kind of need in development another module in this FragPipe-Analyst to look into the protein quantification, and they will only relies on identified data. So through this kind of lollipop plot or like filter and a Venn diagram, we are able to reproduce some of the candidate can only be found in the CCND1 weight protein sample, and this 24 also kind of overlay with early candidate. So this is just another way for people to do analysis. So here is now I also need to switch the topic to the other one. This LibMS. LibMS is an example of a peptide level analysis. This is the general procedure, and the kind of core concept of this part is when you use LibMS, we want to understand some protein target if they are influenced by perturbation. For example, if you have a drug add into a sample, if they can cause some structural change of the protein. So the reason this kind of thing work is we will use one kind of a protease. This protease is not really specific to like tryptic or the enzyme, those things. They are unspecific, but they only cut the peptide out of a protein because they are accessible. So we are able to see if those are accessible and flexible region will be changing before the treatment and control samples. So here is a sample we got it from our collaborator and previous publication. So in this work, they are interested in one of a drug called rapamycin. This is an mTOR inhibitor, and they wonder if there are some protein target will be changed due to this perturbation. So in their experimental design, they will have a rapamycin treatment group and also the control group, and then they will independently go through the LibMS protocol. So after you submit, again, after you submit the protein quantification table, peptide quantification table in this case, since we want to understand if their particular region on one protein target being influenced by this perturbation. So we will use the peptide level quantification table and experimental annotation together, submit to a FragPipe-Analyst, and then we can get a similar result similarly. And then here is the thing we show in the left-hand side. In the original study, they also show kind of similar panel. One thing we want to kind of improve is you might interest in one peptide since they are upregulated, but you want to see how much changes being introduced in other peptide come from the same protein since they are the same protein target. So we actually make this functionality like intuitive one. You click one of a peptide feature, then they will highlight other feature come from the same protein together, kind of replicate the analysis they performed in the original manuscript. And we look into a particular protein target called FKBP1A. That's one of the target protein of interest. So based on our FragPipe-Analyst result, this protein sequence changes between the treatment versus control is kind of similar to what reported in the original study. Unfortunately, we are not able to do this side by side, so it's kind of you need to kind of visually to see some of the region is the same. They use a different protein search engine, so it's kind of expected you will see some discordance in some of the region, particularly on this part of the protein sequence. So now we are finally here, the final example for showing the FragPipe-Analyst or more for the FragPipe-AnalystR. So when we develop the FragPipe-Analyst, I think the general mindset is we want to help the user without the programming background. But as a bioinformatics student, I find lots of things I want to put into a publication need to be replied several times. So if we are able to generate a kind of more like a mature software product, like a package that might be better. So that's a way that I further, after I develop the web interface, I focus on FragPipe-AnalystR is our package. And one benefit that if you can program, you can actually change some of the order of this procedure, since for different application, you might have other interests and you might apply additional filtering. So we make this plugin like modular, so you can switch back and forth, just through programming. But in the original FragPipe, you are only able to define a fixed pipeline that probably most people will do that, but in your application, it probably doesn't work. And one of the interesting things is during the site-specific PTM analysis, it's kind of increasing, got attention, so we want to uncover some of the analysis. So here I will take a site-specific protein population as an example. In this part, we kind of design further module. We can take a site-specific protein phosphorylation result and join together with the global protein estimation together to do an analysis. You can still perform a differential expression analysis, but further, since they are site-specific, we can take particular phospho site and their fold change and submit those into an enrichment analysis, and then get a result that if there are some kinase of interest shows up because we observe those phosphorylation sites. And the reason we want to apply this kind of a protein data together is since we noticed that if you just do analysis on original without any normalization against the protein abundance, the fold change will basically follow the fold change of site-specific phosphorylation will kind of follow global protein abundance. It's kind of expected because if you have more protein, even though they don't have much phosphorylation, they still have a higher chance to get to a method analyzer and get analyzed. So parent protein abundance will largely bias your quantification result of the phosphorylation. So here we will do a simple regression to kind of correct those correlation between the protein and phosphorylation. And after normalize, you can see the fold change between tumor versus normal. They are no longer correlate. So we think that it might be better to capture some of the biological signal. But generally, I think doing this such analysis, we will suggest you need to do both, since some of them, although it's kind of generated by the protein, parent protein aberration, it still might have a biological effect. So here is like an overview. If you submit those things to a FragPipe-Analyst, we can still do like PCA analysis. We compare before and after normalization. They are really still like well separated, even we remove those protein abundance changes. So they suggest they are really active protein phosphorylation and might influence on cell signaling in the cancer versus normal in this case. And similar functionality as I demonstrated earlier, but now you can do it like in a script that you can pull out some of a protein marker previously proposed and see if they are really differentially expressed between the conditions. So as you can see on the left-hand side box plot, so some of the protein phosphorylation marker report in the earlier cell manuscript seems to like have a differential expression result in the original analysis. But if you remove those protein abundance, some of the sites will no longer become like differential expressed. But some of the sites still of interest since after removing the protein abundance, they are still really differential expressed. And we think they might indicate some of the different regulation on those sites and might be of interest to further into it. And as last part, after we get the differential expression analysis result on the protein phosphorylation, we can submit into a site-specific enrichment tool. For now, we only focus on phosphorylation since not many other PTM have this site-specific enrichment tool we can use. But here we demonstrate we can use two different tools, and then they have a pretty similar result and really comparable result. As I show here, we incorporate some of the visualization, but some of the candidate like AKT2 shows are consistent across the two different software. They have a different enrichment procedure, but overall we find this AKT2 is of interest. And when we go back to the literature, it's really interesting. This AKT2 is also reported in other a renal cell carcinoma study. So we kind of think that it's kind of the interest, and we might have further other project to look into in this detail. So till now, I kind of demonstrate all of the 5 examples we incorporate in the manuscript, and I want to make a summary here that we present a downstream analysis tool FragPipe-Analyst for the FragPipe user community to seamlessly perform downstream analysis after running a FragPipe. And FragPipe-Analyst support all major quantification workflow, including the DDA and DIA label-free quantification, and also TNT, and over a range of functionality, including like exploration and even like peptide level analysis. And in addition to FragPipe-Analyst, I also developed a package called FragPipeAnalystR that could basically cover all of the functionality of a FragPipe-Analyst and support site-specific PTM enrichment analysis. We are still actively working on it, but I think we are really getting there. And there are many future work. For example, like feature as a group, we are also having a whole facility. We have lots of requests for generate a quality control report. So I'm still working on generating some module that can generate those QC report like concisely and more peptide level analysis. Since we noticed that chemo proteomics field also have much more peptide level analysis we want to achieve, and including the immunopeptide analysis and more refined PTM analysis for PTM other than phosphorylation also of interest. And in addition to proteomics, some of the project I mentioned earlier, like non-CCRCC or lung adenocarcinoma, is generally speaking, is also a proteogenomics project. All that means is it has a genomics and transcriptomics data, so we want to have some integration together with those data. And in the operation side, we also get a request from the Galaxy group that we might incorporate our software tool in the Galaxy server. Is another big bioinformatics suite. And scaling the server for more requests is also one of the top priority item in my list, since after the publication, I think user crash server a lot, like I need to fix every week. Yeah, but so in the last slide, I want to acknowledge all the people, not only the DCMB, but also the my dissertation committee, Alexi Lab, and the UMH PhD Tech Group, and also many CV Tech Group. We focus on many different cancers at the same time, and every working group we have so many collaborators are not listed here. And but thanks everyone for helping on this project. And so here is all my presentation. Thank you for listening, and feel free to ask any question. And I also put my FragPipe-Analyst tool link here. And happy Halloween.
Thank you for your nice talk. I'm just curious about the potential of the analysis for identifying isoforms or cancer neoantigens.
You mean like in this publication or
Yeah.
or proteomics general?
The pipeline.
So, well, I mean, we did identify different peptide isoform, possibly through the proteomics data, but it really depends on how much you think that's real. And if you want to identify isoform, then you need to put the isoform into the database. But for in this sample, we use the database like reduce a lot of isoforms. So generally speaking, we can find those things. And neoantigen is based on, like I think I mentioned here, if you want to find a neoantigen in the proteomics part, we will use like immunopeptide experience. And in that part, the quantification or database building is also different, since general global proteomics data is showing here, no matter it's global proteome or phosphorylation, they will be attributed digest. But for immunopeptide data, they are more like directly enriched and not really cutting by different enzyme. So it's a specific search. So but our FragPipe have the ability to do that, but for downstream analysis, I think it's still actively developing. In one of my cancer projects, I also work on some HOA data. So if you have further questions, we can discuss later, like how to use our tool to do analysis.
I see.
Gilbert Omenn has a question. Let me just put them. Gil, you
Hear me
you
now?
talk-
Can you hear me?
Yeah,
Yeah.
yeah, I can hear you.
So very nice presentation, and I commend you for staying well within your time too. So I hope you stimulate a lot of questions. I'm interested, you've done so much work on phosphorylation, but we do have data sets, mostly from our collaborating labs, on glycosylation, acetylation, ubiquitinylation, maybe other things still in the works. So for the PTM analysis, where do you see the most interesting work going next? And what are you specifically working on? I take it your R package is especially suited for PTM studies. Is that right?
Yeah, I think that's a very good question. For me, I think glycosylation will be the most interesting one since compared to other examples you mentioned, like acetylation or ubiquitination, they are another layer of the complexity since not only a site-specific PTM, like glycosylation, they will multiply on a site, but even on a site, they have so many different complexity layer. You can have a longer glycan or shorter glycan. Although in this workflow, most of the CPT protocol, we are only able to get a composition, but even a different composition on the single site, I think most of people are not using it for large scale analysis. So I think for our package development, I will focus on this first since they also kind of explore another layer of complexity compared to other PTM.
Yeah. And which labs have made the most special uses, new uses of FragPipe
I think
pipeline?
you mean like FragPipe-Analyst or FragPipe?
FragPipe pipeline, yeah.
Yeah, I mean, in my understanding, I would say probably chemical proteomics field. So that's why I did some of like peptide level analysis, like cysteine level in reactive amino acid residue. I think it's one active region like of interest, and some of the ubiquitination work also compare us to do a thing. So I think that's a major user. But HOA from the cancer or other disease, I think there's another big user community, and there's something we want to aim for.
Very good, thanks.
Yeah, thank you, Gil.
Very nice talk, and I'm curious about the post-translation modifications. As you know, the transcript data and the proteomics data, they are sometimes not really correlated because of some translation modifications are being regulated or degraded.
Yeah.
So in your tools, is there any single or some cutoff, some you could identify some single of the modifications of the transcript?
Oh, I think that's an interesting idea. I think I have a kind of module, but it's a little bit different from you are asking. I think you are asking like kind of like discordant between like post-translational modification side, the SMA at the protein level compared to like RNA quantification. But I think there is a fundamental thing is RNA and protein, they are also not correlate. For example, recently we started doing some analysis in melanoma. Melanoma is a cancer type with high tumor purity. Their protein RNA quantification correlation is really good, probably 0.6, 0.7 in terms of a Pearson correlation. But if you look at prostate cancer, the correlation could go down like 0.1. So even before go to the PTM level, I think the regulation between the RNA and protein will go to the like degradation that you will not estimate at RNA level. I think this original is already a big difference. So I think we have a correlation analysis for module for lab, but go to a PTM side, I think that's a little bit far. I think I can think about it, but for now I was thinking like even protein level, not even like site level will be something of your interest. But degradation, the ubiquitation is definitely play a role. I think you might want to like further look into it.
Thank you.
Yeah. Any other questions?
Not seeing or hearing any other questions, so let's thank you one more time. Thank you all for coming.